And the next speaker will be Noa Eren, from the Weizmann Institute. She will talk about complex behavioral phenotyping in FMR1 knockout mice. Noa, please.
Thank you, Shlomo, and thank you to the organizing committee. So my name is Noa. I'm a PhD candidate in the lab of Professor Ron Chen at Weizmann. And I'm going to show you today some findings from an ongoing project focused on complex behavioral phenotyping in FMR1 knockout mice. So in our lab, we've been focused for the last several years on trying to address a problem in the field of behavioral neuroscience, which is that the classical and commonly used behavioral paradigms are quite reductive and simplistic. So they take place in short time scales. They usually use only one animal or a pair at most, and they employ environments which are usually unfamiliar to the animals and quite poor in stimulation, all with the goal of eliciting a specific behavior or set of behaviors which are of interest to the researcher. And back in the day when behaviors were scored manually, this made a lot of sense. But now with advances in computer vision and data analysis tools, we can use them to expand the limits of these classical phenotyping methods. A second problem, which I'm sure many of you are familiar with, is that autism is a very heterogeneous group of disorders at multiple levels, from etiology to phenotypic presentation. And if we put this together with the limits of classical behavioral paradigms and preclinical findings being themselves quite variable, we get quite a large gap from preclinical studies to findings that are actually translatable to humans and have therapeutic potential. So in our lab, we're concerned in general with the question of how we can bridge this gap from behavioral studies in mice to translational and clinically relevant findings for psychiatric and neurodevelopmental disorders. So the solution that we've come up with is this experimental paradigm, which we call the social box, that's been developed in our lab over the last few years. I'm going to briefly show you in this video what it looks like. So the social box is basically a large arena with some environmental enrichment in which we house groups of mice for days or even weeks at a time. And we record the mice behaving freely in the arena, and we can automatically track each individual by coat color. And from the tracking, we can extract dozens of different behavioral readouts. So the idea is to have a more naturalistic way to phenotype behavior in a social context and with minimal disruption to the animals. So in this project, we wanted to use the social box system to characterize a few well-established mouse models of ASD in a more naturalistic social setting to try to get a more robust and holistic behavioral profile. So as I said, we are working on several different mouse lines in this project. I'm going to focus today on results from one of them, the FMR1 knockout line. So this is a model of Fragile X syndrome that also has a high penetrance of autism. And this mouse model has been around since the 90s and is one of the most widely studied mouse models in the field. And the problem is that in different labs and different behavioral protocols, these mice have shown some pretty big variations in the social behavior domain, as you can see here from this review. So a lot of studies report a reduction in certain social behaviors, while others found no genotype differences. And a couple studies actually reported an increase in certain social behaviors. On the other hand, in the repetitive behavior domain, the consensus is that these mice display an increase in repetitive behaviors. So in this experiment, which is a collaboration with another PhD student in the lab, Stoyakova Michalev, we included homogeneous groups in which all mice are either knockout or wild type, as well as mixed groups that have 2 mice of each genotype, and the ends here are the group numbers. And we group the mice at weaning age, and we house them together in the same groups until the beginning of the experiment. We paint them, and then they go into the social boxes for 8 days. The first 4 days are the baseline stage, where the mice are completely undisturbed. And we also included a stress stage, since we were curious to see how the knockout mice would respond to a stress conspecific. This part is still being analyzed, so today I'm only focusing on results from the baseline stage. And we chose 36 behavioral readouts to analyze that are related to activity, exploration, social interaction, and food and water. So at first, we wanted to examine the general differences between genotypes across the entire set of behaviors that were analyzed. So to do this, we take the 36-dimensional space of all behaviors, and we look at the mean Euclidean distance in this space between wild-type and knockout mice. And we generate a null distribution of what this distance would look like if genotype didn't affect behavior by shuffling the genotype labels on the actual data and repeating it 100,000 times. And then we see where the actual observed distance, which is the red dotted lines in each panel, falls on this null distribution. So we analyze this separately for homogeneous and mixed groups. And what we can see is that while in the homogeneous groups on the top row, the actual distance between wild type and knockout on each day is significantly larger than expected by chance. In the mixed groups, this difference was not significant. So this is our first clue that the makeup of the group can actually affect the mice's behavior. So what about specific behaviors that are of translational relevance? So hyperactivity is a common symptom in Fragile X syndrome, and it's also commonly reported for FMR1 knockout mice. So to assess this, we looked at the total distance that the mice travel in the arena and their median daily speed, and we averaged these values over the 4 days, baseline days. So in the hyperactivity domain, it looks like the FMR1 knockout mice in blue were hyperactive compared to wild type only in the mixed groups, but not in the homogeneous ones. To address the domain of repetitive behavior, we use a measure of mutual information that estimates how predictably the mice explored the arena. So for this analysis, the arena is divided into a 6x6 grid, and the probability of a mouse within a given grid cell to move into each of the adjacent cells is then computed. And using this measure, we found that the FMR1 knockout mice explored more predictably compared to wild type only when they're in homogeneous groups. We also examined social behavior. So when we looked at rates of approaches and contacts normalized to the time outside the nest, there were significant or near significant main effects for genotype and group type in both measures, but the post hoc comparisons did not show significant genotype differences. From the plots, you can see that in general, the knockout mice tend to score slightly higher on these measures. So to summarize the findings so far, we've seen that general genotype differences between FMR1 knockout mice and wild-type controls are present when the mice are housed in homogeneous groups and are absent in mixed groups. We also saw that in specific behaviors of interest, the group composition seems to affect them in different directions. So knockout mice were hyperactive in mixed groups, but showed reduced behavioral flexibility in homogeneous groups. And finally, we find no evidence of a social deficit. If anything, the FMR1 knockout mice are slightly more sociable as they tend to initiate more contacts. So what's next for this project? We are working on expanding this analysis to additional mouse lines, which will allow us to ask whether the importance of the social environment is generalizable to different ASD models, and also to try to identify a common behavioral signature to ASD-like mice. And what I hope I was able to convince you in this short talk is the importance of including more complex, prolonged, and natural behavioral phenotyping methods, and also putting more emphasis on the social context, which we hope together will eventually help to improve the translational value of these preclinical models. So thanks to everyone in the lab who took part in this, thanks to the organizing committee, and thank you all for listening. And these are a few select studies with the social box system if you're interested in reading more.
And thank you, Noa, for fascinating lecture.